Hello, my name is Ayla Zhang, and today I'll be talking about my project, Novel ChatGPT-4 Evaluation Method Reveals SRC as a Promising Pancreatic Cancer Target. Pancreatic adenocarcinoma, or PAAD, is a highly aggressive solid tumor that accounts for over 90% of all pancreatic cancers. And PAAD is distinct from pancreatic ductal adenocarcinoma, or PDAC, which is a common subtype of PAAD, although these two names are sometimes used interchangeably. And PAAD has a very low 5-year survival rate at less than 10%, and is currently the third deadliest cancer in the United States. PAAD is also characterized by poor treatment options. Surgery is the only option with proven success, but since PAAD has very poor prognosis, at the time of diagnosis, over 80% of patients experience metastasis, rendering them ineligible for surgery. Current drugs target frequently mutated genes such as KRAS, but have had low efficacy in clinical trials as well. However, recent findings suggest that SRC could be a promising target for PAAD treatment due to its role in signaling pathways that enhance tumor growth and metastasis. SRC is a non-receptor tyrosine kinase, which interacts with receptor tyrosine kinases, G-protein-coupled receptors, integrins, and more, and is involved in several signaling pathways that facilitate growth and metastasis of PAAD. SRC has also been found to be frequently overexpressed in PAAD. SRC has been targeted by several existing small molecule drugs, which indicates its ability to bind to small molecules. However, no existing drugs target SRC to treat pancreatic cancer specifically. As you can see in this word cloud illustrating the association of SRC to different cancer types based on clinical evidence, current drugs targeting SRC typically treat leukemia and lymphoma. Therefore, the goal of this research was to demonstrate that SRC is a gene that has the potential to become a drug target for pancreatic cancer treatment. And our criteria for this is that SRC's involvement in pancreatic cancer is based on gene expression, it is significant in metastatic or late-stage cancer, and that it impacts patient survival. So in order to prepare a list of candidate drug targets which we could compare SRC against, we compiled an initial 253 gene list from literature on pancreatic cancer based on literature coverage, clinical trials, and expression across tissues. We then narrowed this candidate list down to 30 genes by sorting the genes based on the Beer ranking score, which evaluates and prioritizes a list of genes based on their network interactions. Additionally, in preparation for our next step, we retrieved the top 10 overexpressed genes in pancreatic adenocarcinoma from ULCAN. Then, for each candidate from the final 30 gene list, we calculated a gene score. To do this, we first obtained a pathway list from Pager 3.0 for each of these candidates by inputting the candidate plus the top 10 overexpressed genes in pancreatic adenocarcinoma. We prompted ChatGPT-4 to create a comprehensive evaluation framework to assess the importance and influence of a given biological pathway in the scope of pancreatic cancer, and then prompted ChatGPT-4 to score each pathway based on this framework. Our ChatGPT-4 generated evaluation framework ranked each pathway from 1 to 5, with 1 being the worst, in 6 different categories: pathway relevance, target drugability, genetic alterations, biomarker availability, evidence from literature, and safety profile. ChatGPT-4 ranked each category as a high or moderate priority, assigning pathway relevance and target drugability as highly weighted categories and the remaining categories as moderately weighted. ChatGPT-4's assigned score 1 through 5 in each category was multiplied by a corresponding weight. Highly weighted categories were multiplied by 5, while moderate was multiplied by 3. The sum of the resulting 6 scores produced the total weighted score for each pathway, and the average of these total scores for all the pathways was taken to obtain a final score for each gene. For our validation, we used ULCAN to retrieve expression data and survival curves. ULCAN uses TCGA data and uses the median as a cutoff between low and high expression for the survival curves. We also use the Kaplan-Meier plotter tool to compare survival for different tumor stages. This tool automatically selects the cutoff between low and high expression based on the most significant results. Here's a summary of the datasets we used to perform the survival analyses. The KMP dataset is a Kaplan-Meier plotter dataset, which we also split into subsets of samples with tumor stages 1 and 2, as well as 3 and 4. After compiling our candidate drug targets and ranking based on beer ranking scores, we narrowed our list down to 30 final genes, and this table displays our top 10 gene candidates. In this list, SRC was ranked 2 out of 30. This table shows an example of how we employed ChatGPT-4 to rank each of our gene candidates. After using Pager to generate the pathway list, each pathway, for example, cell-to-cell adhesion signaling, was scored in these 6 categories, which were multiplied by their corresponding weight. The average total for all the pathways for that gene was the gene's final score. This table displays the top 10 genes after our AI ranking, and SRC had the 5th highest score out of our 30 candidates. In order to validate these findings, we retrieved these graphs from YALCAN, illustrating the expression of SRC in PAAD based on tumor grade, individual cancer stage, and sample type. These graphs indicate that SRC is more expressed in tumors than normal samples, especially in later grades and stages. Next, our overall survival analysis conducted on the KMP dataset was significant with a p-value of 0.00769, indicating SRC's significance in PAAD patient survival. Additionally, our disease-free survival analysis was also highly significant, suggesting that high levels of SRC are associated with a more aggressive form of PAD, which may also recur earlier after treatment. Comparing tumor stages 1 and 2 against 3 and 4 also shows that SRC was more significant in the dataset with later stages. Finally, our survival curve of the TCGA dataset, which did not automate the cutoff value, was insignificant, perhaps indicating the importance of dynamic cutoff selection. In conclusion, our work supports the candidacy of SRC as a target for PAAD treatment. SRC was shown to be more significant to survival in later tumor stages, perhaps indicating its potential as a combination drug in the 2nd or 3rd line of therapy. Also, combining bioinformatic pathway analysis with ChatGPT-4 revealed significant insights. Future directions include checking SRC's expression along PAAD progression and designing experiments to inhibit advanced stages of PAAD. We would like to thank Adrian Gu of AlphaMind Club for preparing part of the drug candidate dataset. Thank you.